No, they came to the last meeting.
There are so many friends in this room. Hey, some of you I haven't hugged yet, and I could just stop everything and go around and hug you all, but I'm not going to do that. Some of you I haven't met, but I'm going to, so we're all going to. There's a lot of time after today. So today is kind of an intensive time for us with 3 of these sessions today. A BPAN we've done, a PKAN now, and a PLAN to follow. So it's kind of a killer day for us, but we're looking forward to spending time with each and every one of you. So thank you for being here. I'm going to start off, and then I'm going to quickly hand the reins to Penny to talk to you about COASY. But I'm going to just start off by, again, welcoming you and telling you that we are working so hard in ways you cannot see every single day. And there's nobody here working harder than Penny. And so if you want to take a swing at anybody because you're frustrated, which I know you are, because we are too, we are not the ones to take swings at. What we're going to do in ways that both here at the meeting and to follow in the next few days and weeks is to invite you to join us in taking swings at the places where we think swings need to be taken. We're going to try and mobilize ourselves. I'm counting everybody in this room and everybody who's not in this room who's a family affected by this terrible disease to join us so that we have a common voice that has a force that we need to have in order to get the regulatory body to do what we need them to do. So that's where we're headed with you. So again, I know you're frustrated in coming here. I've sensed that, I've heard that directly, and we feel it as well. But I'm going to invite you to shoot your darts at me and not Penny. I'm a bigger target anyway, so I'll be easier to hit. Yeah. So maybe I'll just stop there and.
Okay.
Yeah.
So much you can stand up for this, being the midget that I am. So, first thing I wanted to ask, and I will admit this is a little bit of paranoia on my part, is who in the room is not either someone with PKAN or someone directly related to PKAN? And I'm asking everyone in the room to just raise their hands if you don't have PKAN or you're not directly related. I know you, I know you, I know you, I know you. I don't know you in the back. Okay, welcome. And was there someone else up here? Oh, I know you. Okay, so with that, I'd like to sincerely ask that what we talk about in this room, stay in this room, just for this session. So I am going to tell you a little bit about where we're at, where we're not at, what's been transpiring, what is coming. So you've all been hearing about COASY for too long, waiting for this study to start, and we have made the most astonishing progress, and we are so ready to start this study. You cannot believe how ready we are. And despite what Susan said, I actually don't take credit for a great deal of it. Allison, the rest of our team at OHSU have been working incredibly hard to put all of the infrastructure in place to have everything poised and ready to go in the US and Canada. We'll talk about other countries in a minute. So where we are at is we are basically ready to start the clinical trial. We have incredibly pure compound ready to be shipped. In fact, right before I walked down here, I was emailing the shipping agent around that. So the compound is ready to be shipped. We have it poised, ready to be made up, not into grape or apple as I originally told you 2 years ago, but into strawberry-flavored syrup. It's now going to be strawberry-flavored. So we really do have everything down to the size of the bottle, the adapter that you'll draw the stuff up into, what type of plastic it is, and, every microscopic detail you can possibly imagine. The database built to FDA compliant status is done. Everything is ready to go. What we are waiting for is for the FDA to make one key decision. It has been stalled for longer than I care to admit. And we are now kind of pushing the FDA. Some of the delay was during the shutdown. And then I expected to get that decision very quickly after the shutdown ended. Didn't happen, and it's become increasingly difficult to get any kind of information from the FDA. We do now, I think, have a pathway to someone, a different pathway to try to push them to make that decision without stepping on too many toes.
You've had that for about 2 hours?
For about 2 hours. So at, let's see, what was it, local time? I think it was 1:30 last night. I was emailing with somebody at the FDA that I had finally made contact to be able to take this through a different pathway to the people that need to actually make what is a very simple decision. So if you're frustrated, I am just as frustrated. I wouldn't say my frustration is greater than yours because I know you feel it in a very real way that I can never really feel it, except that all your children are my children, and I really do mean that. So we're really ready. We are really ready to start this. One thing, just to bring everybody down, I think because of this wait, anticipation, all of the discussion about it, there's a huge amount of hope about this compound. I do want to give you all a reality check. We have no idea at this point whether it is going to make a difference to this disease. We have really kind of compelling and beautiful evidence from a mouse, but a mouse is not a human. Some people in this room, some children and parents in this room have had their disease in place for a long time, and we don't know whether this compound has the ability to turn a disease around, whether it will stop progression, whether it will not work at all. We really don't know that. That's why we have to do a clinical trial. So I want to be transparent with you that even though there's this enormous hope, and we, I know, have fueled that with our own excitement about the disorder, about the compound, I mean, we've got to be realistic that we don't know if it is going to turn things around. So with all of that, my hope is still very much there. So I'm going to open it to specific questions from families.
I want to just make one other comment. So Penny said it's one specific decision. It's actually it's what's called a determination in FDA parlance, and it's actually a series of decisions that they need to make around chemistry, around stability. It's sort of a complex set of decisions, but it's a single determination that we're looking for from them. And so, you know, we don't want to get into the specific details about what the deal is there. All we know is that we're hung up, and it's not clear to us we're hung up because they're working diligently day and night to get to an answer. It's gone into a little bit of a black hole, and that's part of the source of our own frustration, is that we haven't been able to find the name of somebody working at the FDA whose salary is paid by you and me, who can tell us where it is, why we don't have an answer, when we will have an answer, what's a realistic time frame. We don't have that information, and so it's, as much as anything, it's this kind of absence of information that is so frustrating to us.
Yeah, just trying to get any information. We had a really great advocate at the FDA with whom we've been working for the last 3 years. She got promoted, which, if it had been a promotion that might have helped us, I would have celebrated, but it sort of took her out of our realm. And so we're working with a new group of people, which I think is one of the challenges, but that isn't the whole story. That's just one little piece. And So that's where we're at. I am living and breathing COASY when I'm not talking to BPANers. So I do want to say, because I see faces from Australia, I know we have some people from Europe here, from the UK. So for each one of those places, for Europe, the UK, and Australia, we have active conversations going with parties in those countries to be able to extend the trial as swiftly as we can into Europe. Europe is challenging because of the way the regulations work between countries. And so it may be that it has to roll out kind of country by country in Europe, but there are, and I'm looking at you too, there are regulations that are EU-wide that will help us. And so we're still navigating exactly how it is going to play out in Europe, but we are actively talking about the study starting in the European Union. Because of Brexit, who the hell knows what's happening in the UK, but we are actively talking with our colleagues at UCL, at University College London, and then also with a group in Australia to try to extend the clinical trial into Australia. So, you know, we're doing everything in parallel. I sometimes feel like, you know, I'm just sweeping every little bit forward, one little piece at a time. But the overall trajectory is unerringly forward. And I want you to hear that, despite our frustration and your shared frustration, your much more meaningful and poignant frustration because of what you are experiencing yourself or seeing in your children. So please ask your questions.
I'm going to jump in for a second, and actually I want to talk about a couple of other things that might spur some questions, and then we'll have plenty of time to talk about specific questions that you have. So, you know, we both are measured in what we're expecting of this compound, because the truth is that nobody knows. But Penny made reference to the mouse data, and you know, even after we saw you 2 years ago and have continued the work in the laboratory, the mouse data, every question we asked, every way we could think to ask the question, is this changing the biochemistry in the mouse? The answer was yes, and it was changing it back to a mouse that doesn't have a mutation in PANC2. It made the changes that we saw look like a healthy mouse. We don't see a sick mouse. We see biochemical changes in the mouse, but the compound was sort of correcting the primary problem in the pathway in such a way that we saw improvement not just in the pathway itself, the B5 pantothenate pathway, but we also saw normalization or correction in the iron management and in the nerve health, in the chemicals that reflect nerve health. And it was really those changes that were the most compelling to me, to see that we could feed the mouse this compound, the PKAN mouse, this compound, see those chemicals normalize, stop the compound, see those chemicals reveal the disease process. So this is what we do. We're scientists. We write things up. We publish, we make public our work. We do that for a number of reasons. In order for people to believe us, right? You trust us, not without good reason. We've done good work through the years, but the way a scientist has their work criticized is by putting it in the public domain for other scientists to look and say, well, you're concluding that, but there's 4 other ways to conclude it. Or, I see your data, that's really impressive. And I will tell you that I've presented the data, admittedly with a lot of enthusiasm, to the child neurology community in Barcelona in February, and to the pediatric metabolic community just about 2 months ago, and they were extremely excited about it. So I also presented a little bit of this data at the child neurology meetings in Mumbai in February, or in November. Yeah, we have a paper actively under review. So talking about papers under review, there's somebody here who is not showing up on the screen. I had a really good animation. So lurking in the audience is Sue Youngjiang. So Sue, put your hand up. Yeah, so that's Sue. Sue is the lead in our lab. She's a scientist, a PhD scientist, trained in Canada. She did a postdoc with a very prominent iron biologist at the NIH, and then we were fortunate enough to snag her out in Oregon, and she's been working in our group. Sue gets a whole lot of credit for creating the mouse model, which was essential for us to be able to see whether CoAZ was doing what it needed to do. And so the picture's not coming up, much to my dismay, but Sue's here. I would encourage you to, today's kind of a busy day for our whole team, but encourage you to pull Sue aside and feel free to thank her, but also to talk to her about her work, because she's been doing just amazing work. So I just wanted to call her out. Yeah. Yeah.
So having given you all a sort of high-level frustration, now I want to talk about a little bit of the nitty-gritty. So I think many of you know this, but I'm going to be explicit in it. This trial is not going to be like other clinical trials where you travel to a study center, you get seen by study doctors, you get your blood drawn. We are trying very, very hard to make this as easy as possible for families to participate in their own community and not have to travel. The reasons for this are many. If you have an ultra-rare disease like PKAN, for almost everybody, travel means a long way. And particularly for the children with classic BPAN, they don't always do so well with travel. And even some adults, they decompensate, they just don't do so well with long travel in all cases. And so for us to run a clinical trial from Portland and have everybody fly to Portland, it sounds easy in concept, right? Just throw money at it, you make it happen, but we're not going to do it that way. We want very much for this to be a novel clinical trial design where all of the activities happen in your own community. We have worked very hard to get a contract negotiated with Quest Laboratories, who will be doing the blood draws for most people, and for those few families who don't have a Quest anywhere near them, we're making individualized arrangements for blood draws that will be needed through the clinical trial. You'll complete online assessments. The product will be mailed to you along with kits to get the blood draws done. We will coordinate everything through your local community. You, as participants in the clinical trial, will have a set of responsibilities that we're going to hold you to. Getting blood drawn on time, completing some diaries. It's nothing very onerous, but we absolutely have to know that the compound is safe. That is the primary goal in this first study. And in order to do that, we have to be able to kind of look at your blood samples over time. And so we're going to make it as easy as we possibly can for you to get those blood samples done. But this is not a clinical trial like most clinical trials where the burden is all on the family to travel, to get everything done, to kind of manipulate their schedule, to make it work with the study center's schedule. We're trying to make it as easy as possible. So it's an unusual clinical trial. Even for people who live in places where blood draws are going to be challenging, we've got you in mind. We've got your interests at heart here. So, okay, on to other things.
Well, not necessarily. What I wanted to do was actually go through the few questions that were submitted previously and then open it up to people to talk, to answer your questions. 2 points came up when Penny was talking. It's actually not a U.S. trial, it's a North American trial. we think of Canada as the far north of the U.S. Actually, we think of the U.S., I wish we were the southern part of Canada sometimes.
Especially
So
right
that's
now.
one part. It's actually, it is North America. It's in theory, it's Mexico as well. The other piece I wanted to just comment on is, so many of you have been front and center in the fundraising effort that's gone on with Spoonbill, and I want to acknowledge that. You have been nothing short of amazing. Whether it's sort of in small ways with a lemonade stand in a rural part of Oregon raising $2.80, damn it, that is good work, and that's contributing to the effort, or whether it's selling a house in Utah. It's amazing. So I just wanted to acknowledge that. I wanted to tell you that without those funds, we would not be where we are today. And so it's not that if we had more today, we would be going faster. We have enough money today to be where we are. That is, funding is not holding us back. The FDA is holding us back. But if we didn't have the Spoonbill funds, we would be a long way back. And so your fundraising has made it possible for us to be up against the FDA door, I would say banging on the FDA door now. But the funding is not now, and really has not at any stage in this project held us back. I wanted to make that clear. Talking about therapeutics, I do want to talk about some other therapeutics. So you all know that we're not involved in the Retrophin trial, but there is an opportunity for families to speak here or in the session Saturday morning. There's an hour-and-a-half session for parallel sessions, disease-specific, for you to talk to yourselves, to each other, about your perceptions in the Retrophin trial. There is a physician from the Retrophin trial who I think is supposed to be here at some point over the weekend, and she can presumably also speak to it. There are other compounds coming. There's at least 2 that are, there's some work going on in, and but the interval to actually reaching clinical trial and whether they will ever reach clinical trial is something I just don't know about. Not because they're not telling me, but because I don't think they know. They're reasonably effective compounds at doing what they were designed to do, but I have a concern that they may have been designed to do something that isn't specifically relevant in PKAN. And I can talk to you about that if you want offline. But that said, there's a lot of activity in this area and a lot of interest. Some of the therapeutics that are being developed in PKAN may have at least a theoretical basis for some of the other NBIA disorders. And so we're exploring whether there might be an opportunity to try out CoAZ or some of the others in other NBIA disorders. So those are kind of the small molecule approaches. We also have begun, because we now have a really good mouse model in the laboratory, the model that Sue developed, we have started work on a gene therapy project, and I wanted just to ask L'Oreal to put her hand up. Yeah, L'Oreal is a scientist. She got her PhD at OHSU, and then she went east to one of the best gene therapy labs in the world. And she's kind of gaining a broadening of her skills in the science around gene therapy. And then she has gotten interested in PKAN and has started to develop some resources to explore a gene therapy approach. So one of the reasons why PKAN is attractive as a gene therapy disease target is that the disease it's just right here and right here. It's in 2 parts of the brain, and when we look at the rest of the brain in a person with PKAN, it's totally normal. And so if we can replace the gene in those regions, kind of like you put a DBS electrode in, but instead of just buzzing it with an electrode, you're actually putting the gene back into the cells via a virus that infects those cells. We think that that's an approach worth exploring. And so we're still early in that, but between Sue's work and L'Oreal's work, we have a team that we think can move that work forward. And so I'm also going to encourage you to find L'Oreal. She's easy to find because she looks like she's from Portland. Now I have to be careful because the Tinks are here, so she also looks like she could be from wherever you guys are from. She's got that kind of easygoing Portland thing. And she's short, that's right. So if you can't see her, just get down kind of low and you'll, yeah. Yeah, and also Sue can talk to you about a little bit more also about how her animal model can be useful for that. In the world of therapeutics, like disease-modifying therapeutics, I think that in my view, that's sort of where we are. It's an incredibly exciting place to be. I know it's not far enough yet. I know that. But it's an incredibly exciting time to be getting on with this. We did have some questions from families about specific disease management, particularly around dystonia. So we field calls almost every month, sometimes more often, from around the world, individuals who are suffering from a sort of sustained severe dystonia that the physicians and family aren't able to manage. We can talk about dystonia management strategies, but it becomes very very, very individualized to the person. We have published a consensus management guideline, and we have included in that guidelines and recommendations that are sort of general for approaching a child or adult with PKAN who is in an extended period of profound, severe, disabling dystonia, which is what a dystonic storm is. It's a little bit hard to go beyond that, though, because your child, your sister, your parent, in some cases, has their own particular needs, and we usually address those needs as they come up. And so we're happy to answer questions if there are questions that are kind of generalizable, but usually they go to the specific child. There was a question about dystonia scales and tracking, and there are scales. There are formalized scales that have been developed in the movement disorders community, and maybe Penny and Jenny can speak to these. I realize I didn't introduce you guys, so I'll do that in just a second. Sorry. I just assumed everybody knew each other. Ask you to kind of talk about those a little bit. We don't, in a patient care kind of orientation, we don't rate the kids over time. That is, we don't do a scale every time you come in. Would it be helpful to? Maybe. I think just kind of talking and getting a sense of how the person is doing is generally more helpful. But let me take a moment and ask you to introduce yourselves. Apologies for that again. And then maybe we can talk about dystonia scales. So Kirsten, yeah?
Hi, everyone. My name is Kirsten Campbell. I am a pediatric palliative care physician. Actually, I'm based here in Charleston at the Medical University of South Carolina. Been a couple other places before that, but it's nice to be here with you all and try to help address any questions that we have today.
Thank you.
I'm Jenny Wilson. I'm a pediatric neurologist at Oregon Health and Science University, and I work with kids with tone and movement problems, kids with developmental disabilities, and see a bunch of other neurologic complaints.
Everybody must know Allison.
I feel like I don't really need to introduce myself. I think almost every one of you know me already. I'm Allison. I'm from OHSU.
So if Jenny were anywhere else in the world, she'd be the local world's expert on PKAN because she has seen 3 patients with it, which is a lot. It's three more. Is it more than that?
Yeah, with us.
Oh yeah, the ones who've come through the clinic. Yeah, of course.
PKAN patients.
Anyway, but she's outfoxed by us. Yeah. So dystonia scales?
Well, first of all, Allison sort of glossed over her role here. So Allison is a genetic counselor, project manager, woman extraordinaire.
Heart and soul of the program.
I mean, so many of you, she has been your frontline person. Susan and I say, and she thinks we're joking, but we're actually not, that if she ever leaves, we're both retiring because we really couldn't do without her. So she's, we got a very nice kind of message recently from a family in Europe who, she was their frontline person. She was the first person that they were able to connect to after the diagnosis, and I think that she has been just the most amazing resource to families. So just to give her credit where credit is due. I'm going to ask her to talk about how we're doing it in the PKAN Ready study, which many of you are already in, and you know what we're doing there. So the dystonia rating scales, which are designed to be completed by a clinician examining a patient, they have limitations in my view, and I'd be interested to hear what Jenny thinks about them. We have tried to apply them to PKAN, including in a clinical trial setting, and we found that they're pretty limited for detecting change over time. They're kind of a blunt instrument to capture some of the true disability that dystonia can cause. And so, we haven't really landed on the perfect tool yet. So what we have chosen to do instead, partly because PKAN is an ultra-rare disease, and so if we're going to collect a clinician-completed scale, you have to make some effort to ensure that clinicians are completing that scale the same way, one to the other. Otherwise, it is really of no use. And that is a pretty challenging task, it turns out. So what we've chosen to do instead is to have a patient-centric natural history study where we are asking families to complete various scales. I'm sure many of you already know about this because you're doing it, about how their disease affects their day-to-day life in a number of different areas. And so that's been our sort of response to the limitations of the rating scales. We certainly welcome receiving information. If a given clinician is completing a scale on a given patient over time, I think that it can be helpful, particularly in kind of acute care management, perhaps in the dystonic storm setting, where some type of a dystonia severity rating scale is being used to sort of see how things are going day to day during an inpatient hospitalization or a particularly severe time. Outside of that, you guys all know dystonia can, vary from one end of the day to the next. It's not clearly always related to where the medications are or, the phase of the moon or who knows what. It just varies a lot. And so I think the dystonia rating scales have been pretty limited. I don't know if you have anything to add there, but.
Yeah, I think I would agree. Most of them kind of look at each body part and then have you grade it on a scale of 1 to 5 or 1 to 10, and then you add up the numbers essentially. There's one that came out kind of recently, which is the Dystonia Severity Action Plan, and it's available online through AACPDM, which is the American Academy of Cerebral Palsy and Developmental Medicine website. And basically, it's like there's 5 levels, and it's kind of more of a functional approach. So the mildest stage is able to sit comfortably, basically, and getting periods of sleep. And then 2 is unable to sit comfortably, can only lay down, maybe having some interruptions with sleep. 3 is can't even lay down comfortably, not sleeping. And then 4 and 5 are basically you're in dystonic storming situation at that point. So they've got a kind of a nice way of looking at it. At least it's more action oriented. Like if you get to the higher stages, we should do something different. But I'm not sure how widely used that is at this point.
So I put up a slide from Allison to talk a little bit about the natural history study. So we've been doing this in parallel with all of the therapeutics work that we're doing, and I will say that it is a question that the FDA asked. Do you have a natural history study? And they were relieved and very enthusiastic to hear that we did. We had natural history data. So the work that you all have been doing over the past however many years, plus the work that Allison and the group have led, Penny's been a big part of that too, has laid the foundation for showing us where the trajectory is without disease-modifying agents. And now, if we have agents that do modify disease, we have a baseline, and not just for COASY, but for other therapeutics to come. Right? The idea is not not to keep this data secret. The idea is to share this data so that it's used by others for purposes of benefiting the community. Do you want to talk a little bit?
Yeah, sure.
Okay, so just to start, I mean, I think all of you who are in PKAN Ready know that you're usually not talking to me about it anymore. You're talking to Allison Freed, the other Allison, or Katrina. So they're kind of the front line on this now. They've figured out good ways to email people on other continents and do it by phone if you're having trouble typing. And yeah, I think we've really tried to be accommodating and make sure that it works for everybody. We have 3 different, what we call Ready studies now. PKAN Ready was the first, and then we did PLAN, and then we did BPAN about a year ago. So PKAN was really the one we learned on because we did it first. So, you know, we really wanted to make it like all automated, and these things were going to be so great, and then the technology didn't always work the way we expected, and sometimes we found it's easier to send people a PDF or do it on the phone or whatever. So thank you for being flexible and working with us as we went through some of those kinks. Because this study's been going for 4 years now, which is amazing. It's a good longitudinal, you know, over time collection of data. Some people have done as many as 7 visits. You all who do them know that you do them every 6 months. So we've accumulated a lot of change over time, seeing how your PKAN has changed in those years. We look at PKAN disease milestones, so those are things specific to PKAN that we thought were really important. When did you get a G-tube? When did somebody first use a wheelchair, need AFOs, that sort of thing. Lots of patient-reported outcome measures that I think parents help a lot with in terms of, you know, how do they see their kids doing with social interactions or anxiety or pain, being able to use their upper extremities, those sorts of things. We get medical records to confirm certain things. That's really trying to be a little more rigorous when we talk about the FDA and what they want for a natural history study. And then we look at developmental milestones, so things like when did you first walk, speak, run, draw a person, that sort of thing. So because this study's been going on the longest, in late 2017, we did a feasibility study with our statistician who's at Washington State University. And he's a guy who's sort of using these progressive new models that Penny understands a lot better than the rest of us, called latent growth curves. He's used them not necessarily in healthcare, but in education and other areas of interest, but they carry over, and they're really nice because you can use them on a group, but also on individuals. And you can almost make like a growth curve for somebody of their PKAN, and then try to extrapolate out to where they might go next, or if you did an intervention, you know, then try to measure the change in that way. So we did this feasibility study just to have him look and see if he really thought he could use these models on this data, if we had enough people, if we had enough data for it to be meaningful, if we had the statistical power, and it came back looking really good. You know, that where we were at then, we weren't quite there, but since then we've collected more data that we should have the statistical power to do what we're hoping to do. So right now we have 77 subjects enrolled. Of those, 28 have done 6 or more visits, which is really quite a bit over 3 years. And then another 25 have done between 3 and 5 visits. And then the rest are just, for the most part, more newly enrolled. They were just diagnosed in the last year or so, so they've done fewer. And then one thing that's really amazing is we did build in an opportunity for people to give us information about their kids who have passed away, because those families still really wanted to participate and be able to contribute. So 6 people have done that, where they do sort of a one-time login and give us a snapshot of where things were at when their child passed away, and then we get medical records so we have an earlier point to compare that to. So we've really appreciated the people who have done that. I think one other thing I might want to point out now that we've been doing this study for 4 years is sometimes we get people who miss visits. That's going to happen. Life happens. Kids are in and out of the hospital. You know, these things happen. Sometimes we get people who I think are a little reluctant, or they get, you know, like all of us, a little fed up with doing these things every 6 months. And they say it's the same set of questions, and they're so depressing because, you know, we're asking these questions for a reason. We're asking the question to try to track progression of disease, but that doesn't mean that answering the questions is easy. Because you're basically logging for us that something new has happened. Somebody had to get a G-tube. Somebody lost the ability to do something. And I really appreciate that, and I really appreciate your willingness to do it anyway. And I also understand if you can't. There have been a few who just say they can't. But we appreciate your help. Yeah.
All right. Okay, I'm going to, I think I'm going to open it because I think that that's really all we had for presenting. We have about half of our time left, I think. Yeah, just about exactly half of our time left. So
The floor is yours.
the session is being recorded, and so if we don't have a mobile mic, so maybe, oh, we do have a mobile mic. So we'd like you to use the mobile mic, or I will repeat your question so that it just gets captured for later. But ask away. The gentleman in the red.
Is that right?
What can we do to help with the FDA logjam? Is there anything we can do? Congressmen, presidents, whatever? Who do we need to write to?
Yeah, so it's we're actually figuring that out right now. So we are talking to a group at the FDA. We've just started a conversation with a group at the FDA that is sort of outside of the usual realm. And I think they are going to advise us on the next best steps. Want to talk about what we have been...
What we're teeing up?
Yeah, and so this has got to be done carefully.
So you'll get two answers. Penny will give you the careful answer, and I'll give you the guns a-blazing answer. Not exactly guns a-blazing, but I mean, she's as impatient with it as I am. She just is more of a rule follower than I am.
Which is not a bad thing, by the way.
Especially when you're dealing with a regulatory agency. They're all about rules. So and again, you know, if you talked to us 2 days ago, it would have been different than 2 days before that and 2 days before that, and we are living and breathing this. So what we're thinking about now is to harness the full force of you all, and the rest of this is going to have to be U.S., because the FDA, sorry, they don't care about Canada and Australia, at least not today in this topic, is to harness the U.S. PKAN population and ask you to write letters and have one person identified in the community. And we would like to actually invisibly orchestrate this so that it's being done in a way that we think will be effective and will help the cause, but not derail it, which is also always a risk with regulatory agencies, but actually to ask you to write letters, to provide us with even a picture of your child 2 years ago, 6 months ago, whatever you think is poignant. Make it electronically, have it be collected by one parent who writes an articulate, forceful, brief, overarching letter that covers it all, and then you all come in. It's like, you know, left hook, right, whatever that is. You know? Yeah, that. Yeah, what that guy in the hat said. That is the hat of wisdom. Yeah, and try and do a 1-2 punch and get them to do the right thing. So we've just come up to that precipice and made the decision that we're going to actually talk to some people here and get some mobilization on that. And if we need it, we'll have it. And if we don't, good, right? So that's what we think you can do. We don't think actually going to your Congress people or higher is the right action now. We think continuing to work within the FDA and to appeal to their humanity, because, I mean, I'll tell you that every meeting that Penny has had over the last 3 years with them, she's taken videos of your kids and she's shown it to them, and they're very impactful. So the people in the room are working, you know, with that information. It's now kind of gone out of the room to other people, and we don't know who they are, what they know, how much they care. And that's part of the problem, is that we don't know that we're like having the right impact on them. We don't know that they know how important their delays are, how much that's a problem. Yeah.
any non-Americans also be involved, like in a way of involving the media to say what is going on in the U.S.?
Yeah, so we don't want the media involved yet, and I'm going to plead with you not to take that step. This is going to be escalated as needed in ways that we think will be effective. Media escalation sometimes is the right action, but in this case, at this moment, we think it's not the right action. It will force them to sort of retreat into their holes, if you will, and defend. And we don't want them to do that. We want them to keep their hearts open to this project because the data are compelling, the information we have is compelling, the plan is compelling. They have everything they need to make these determinations. We just need to get them going. But it's on the list of actions we could take, so thank you. And it's not, I don't want to undervalue the non-US people in the room. You're just as important as everybody else. The FDA cares a little less because you don't pay their salary.
Well, we would be willing to. That would make a difference.
I wish it would, yeah.
You know, and we have given you all this message, and it was not disingenuous. We genuinely have been working very closely and collaboratively with the FDA until relatively recently. And I think the change is simply because this particular set of decisions that they have to make has gone to a new level within the agency. And we are suddenly no longer dealing with the people who have seen the videos of your children and the people that we have been face-to-face with at in-person meetings at the FDA on now 3 occasions. You know, we've gone to the FDA, we've had sit-down meetings with them. They have been unfailingly helpful. So that was not false information we were spreading, you know, when we were telling you that. They really were working collaboratively with us. I think the difference is that this decision of its very nature went to a new place in the agency, and suddenly we have no traction. There's no person to whom we can reach out to, that I know, that I can talk to on the phone. However, last night at 1:30 in the morning, I might have found somebody.
We have some traction now.
Yeah.
At least we think we do. So let us play that out, but hour by hour.
But be on alert, because
Yeah.
we may call you to arms. And
Yeah.
again, as Susan said, I think what we would ask for is a really brief, like 4-sentence summary of what PKAN is for you and your family, and a picture of the person who is affected by it, or 2 pictures.
Other questions? There was one in the back. Yeah.
Well, when you get around to calling congressmen and stuff, our friend actually she's a congresswoman on the Food and Agriculture Committee.
Okay.
So let me know.
Okay.
I can call her.
Yeah,
and
gotcha.
let her know and try
Okay.
to get her to get things going.
So what may be at some point beneficial is not the Congress people publicly calling the FDA out, but a connection like that, walking into the building and talking to people. That actually may be something we tap you for, so thank you for coming forward with that.
We also, in Oregon, we have a very proactive congressman who's been very connected to OHSU, and we are sort of holding that card for, if we don't get anywhere with this latest measure, we have, I think, a strategic plan for what do we do next, what do we do next, and somewhere on that list is Congress.
Yeah.
So.
Yeah.
Keep in mind our friend is in Congress. Elections are 2020, so
Okay.
I have confidence in her ability to get reelected.
Good.
But I can't promise anything.
Okay,
right now, so.
rightio. I gotcha. Yeah, there's timing is always relevant when it comes to politics. So yeah, thank you for that. There was a question in the front. Yeah.
Is there any indication that part of the holdup with the FDA process may be with the new clinical model, being able to have the clinical trials done locally rather than going to those centralized collection points.
Actually, it's not.
Yeah.
That is, in fact, that model was applauded by the FDA. And
Yeah.
so they've, changing things at the FDA is a bit like steering a battleship. It doesn't happen quickly. But they have, in recent years, really shifted to patient-reported outcome measures. And so many of you know that we've submitted federal grants for funding for the clinical trial. And the first grant application that we put in was to the FDA Orphan Products Division. The only part of the FDA that has any grant-making capability is the Orphan Products Division. They have a small grant program. We got absolutely rave reviews, but they didn't fund it. But the thing that they called out in 2 rounds of reviews was they loved the fact that this was a patient-centric, locally done clinical trial with patient-reported outcome measures. So quite the opposite, I think.
And I have one more follow-up question to that. So the last time that you guys posted on the PKAN group was about the shelf life studies, and that those were still in process. And of course, this was before the shutdown. So I was just wondering if you could give us an update on where we're at with the shelf life studies.
I would be glad to. So during the shutdown, so in an ideal world, you submit your shelf life study for review before you do it. This was one of these moments where we had to make a decision. Are we going to wait for the shutdown to end, submit the planned shelf life study, wait for their response, and then start the shelf life study? If we had done that, that would have pushed the start of the clinical trial out even further. So we made the decision, shutdown or no shutdown, we were going to start our shelf life study. So we designed, with your help and your funding, we could not have done this without the funding that came from Spoonbill. Nobody else was paying for this. We put together a Cadillac shelf life study that I think is above reproach. It has been going on. We're just coming up on our 3rd month of beautiful stability data.
Yeah, so we're not waiting, nothing is waiting except for the single set of determinations that we need from them, but we're not, sitting quietly and patiently, waiting for them to tell us, should we do this? We're barreling forward with everything, and as Penny said, we couldn't do it without the Spoonbill funds because there's no other money to support this work. So.
I do want to, having said that, I do want to give a shout out to 2 small grants that we got. The Friends of Dornbecker, we asked many of you to submit fabulous pictures.
Yeah, that was great.
The Friends of Dornbecker are a group of, and I say this with the deepest respect, mostly gray-haired ladies associated with the Dornbecker Children's Hospital who have a small grant program. They gave us a small grant, but it is one of the largest grants that they've ever given to support some of the mouse work. And they also supported at least the start of the shelf life study, but we couldn't have really done the Cadillac study without funding from Spoonbill. And so it was really kind of amazing to do that. And then the other grant-making agency was a small grant from the Collins Foundation. So, yeah.
Yes.
When you were very excitedly telling us about it 2 years ago, you said that the drug would be available easily over the shelf. It wasn't going to take ages to do, and it could be with us in a couple of months without going through all this.
Oh, yeah.
Mm-hmm.
Yeah.
So this is a totally different.
So I was talking about 2 compounds. Yeah. So I was talking about CoAZ, which is a compound that had never been synthesized and given to humans, and a second compound that was an FDA-approved compound. You're making reference to that second compound.
Right, well,
so
where's
let
that
me
one
tell
gone?
you. Yeah, well, I'm not always so clear also, so I may not have been clear. That second compound we tested. So it was an idea based on some data that we had very recently generated in the mouse model. And so this is one of the things that, if we try something in the mouse and it doesn't work, we don't just go to humans and do it. That I can't make sense of that kind of a sequence. What we did was we did it in the mouse. So we had an abnormality in the mouse that suggested that this particular medicine might be able to normalize things in the mouse. We gave the mouse 2 kinds of this class of medications, and we saw that things went, I don't know if they went in the wrong direction, but they didn't go in the right, nothing improved. And Sue's coming up because she did the experiment.
Yes, we checked that in 3 different systems, mouse and cells and one more, and we didn't see the expected data, and actually it was driving the transferrin receptor 1 more, which will bring more iron into the cells. So I have a long cell pathway explanation I can give it to you, but I'll skip that part, and we didn't see the expected outcome.
So again, we initially saw an abnormality in the mouse model that suggested to us that this FDA-approved class of drugs might be an approach. This is separate from CoAZ. This is a whole different topic. And so we tried it in the mouse because it was just an idea. We tried it in the mouse, and it not only didn't correct things, but it suggested that maybe they were going in the wrong direction, and we dropped it. Yeah. So I apologize about that misconception, and that would have made me insanely frustrated also if I thought that Hayflick was sitting on a FDA-approved drug and just not telling you about it. Yeah. Yeah. Rick?
Susan, assuming you found the right person at the FDA and they make their decisions or their determination next week, when would the trial start?
Allison? Well, let's just say.
Give me a worst case.
Well, I might start weeping at this point. So in all honesty, I fully expected to come to this meeting and be consenting people for the study. No kidding. We were teeing everything up so that we could consent anybody that wanted to be in the trial who was at the meeting, do that here. We've given ourselves a 60-day screening window, which won't be that long, but we kind of expect that we're going to get this huge glut of people enrolling as soon as we open it, and we need to collect the genetic information, like it collects the screening blood samples. There's a lot of things that have to happen, so we can't enroll 60 patients on day 1. So we gave ourselves a 60-day screening window. But, to have to give up the opportunity to enroll people at this meeting was really, really a big blow, I've got to say. That said, the database is built. So what has to happen before the clinical trial starts? The stuff has to get to us, the first ½ kilogram of it that is made at this point with greater than 99% purity, by the way. It has to physically get to us, get through all of the regulatory kind of stamps of approval to be released to our research pharmacy. We have to put the final touches on the database, which I think is scheduled by
June 24th.
June 24th. The database has to be fully validated. That is work that is partially funded by funds that you, I think, helped to orchestrate through the NBIA Disorders Association, this FDA-compliant database. It has to be fully validated. These are very mundane, boring things, but they've got to be in place. So June 24th is our target date for that.
One more IRB, passive IRB.
Right. Our study actually has IRB approval. It's had it since January 24th, if you can believe it. But so an IRB is an institutional review board. It is an ethics board that reviews all human research that is done at our institution. So they've given a stamp of approval to our proceeding with the study, and the only item for final approval is that FDA determination. So when we say there's a series of dominoes, that is one of the dominoes. We get the FDA determination, we get our final IRB approval, we are then approved to start human studies. And I think at that point we would open enrollment. We have to formulate the product, so the actual base chemical is coming to us, and we then have to make it up into this strawberry-flavored syrup. We have all the systems in place to do that. We have the team through Oregon State University is going to be doing the formulation work for us. They are totally ready to go. They've got a room separately assigned for this. I mean, I can't tell you how much more ready we are. So I think we would start enrolling people at this point, perhaps by July. Yeah,
Yeah, well, and the other thing I was going to point out is because we know it's coming and because there's certain little parts we can't work on right now, the other thing we've done is nearly all of you are in our repository and or PKAN ready. So when somebody enrolls in this trial, there are going to be certain steps. We have to confirm their diagnosis. We have to make sure we have genetic or medical records proving they really have PKAN. Not because, we know you have PKAN, but if the FDA audits us, we have to prove you have PKAN. So all these kinds of things, like our study coordinators have been delving back into records from 20 years ago, pulling what we're going to need. Like we're getting it all teed up so that it's seamless and efficient the minute we get a go signal. And that's something that we at least have the capability to do right now from our other repositories.
Yeah, question in the back.
I'm sure I probably know the answer, but is this a double-blind placebo study? So how do we,
Yes.
like how long is the
Yeah.
placebo part? And
So it's a double-blind, placebo-controlled trial for 6 months. So enrolled participants, it's a 3-to-1. So three with getting compound, 1 getting placebo. So 3 out of 4 chance of getting compound in those 6 months. And then it extends for 18 months, everybody on compound. So the 3 differences are different dosages. But the starting dose is the dose that corrected the measures fully in the mouse. And we're just looking for that the compound is safe and tolerable through that range of doses, which we have no reason to think it wouldn't be. Yeah.
So the primary question that's being answered by that double-blind phase from a scientific standpoint is to look at the different dose effects, the different doses, both for safety and tolerability, and for their effect on a biomarker in the blood. Now, you might ask, well, why are you doing a double-blind study then if that's all you're trying to answer? Because the FDA wants us to, and because we need to know in a disease that makes a lot of bad stuff happen, we need to know that it's not the drug that is, not the compound itself that is making bad stuff happen. So those of you that have had children who've gone through dystonic crises, you know probably better than anyone else that PKAN is a tough disease, and on any given day, your child can be doing badly or well. And if that is happening in the context of a clinical trial, the first assumption is that anything that goes badly is because of the thing that you just put in your kid's mouth. And so we have to be able to sort out what is PKAN, what is the compound in terms of its safety profile. That's good medical practice, and it is also very much favored by the FDA. But it's 6 months.
Is there an interim analysis?
Mm-hmm.
Do they do an interim analysis?
Yeah, so we are doing what's called an interim analysis for the biomarker portion. So we've structured the database so that we can look at the data after everybody is through the 6 months, and then everybody will go on to the middle of those 3 doses for the remainder of the 18 months. So any other design simple design questions.
So, go ahead.
Not that really is important at this point, but like, what's the dosing schedule looking like for?
It's oral once a day, or per G-tube once a day.
That's it.
Yeah.
Once a day. Wow.
Yeah. And the stuff is delicious. I'm told that some other stuff out there isn't, but so this is. It's not 5 times a day. It's once a day. It's
Yeah.
delicious. I want to give particularly Dr. Campbell a chance to talk. So some of you will get to meet Dr. Wilson, who's the other Oregon physician, at the training session tomorrow. Pediatric palliative care specialists are a very rare breed, and she's come now to talk to all 3 groups today. And I found it very helpful just to hear from her this morning about her view of how palliative care specialists can help. You know, many of us hear the word palliative and think end of life, and particularly for kids, that is not. I want to reframe that a bit. So I maybe ask you just to speak a little bit about your work and how you see your and other palliative care specialists serving families and particularly with PKAN. Yeah.
Sure.
Thank you.
I have to say, I first heard about this conference from a PKAN family that I worked with pretty closely at my prior institution, and it is a lot what you all are sort of dealing with, and I think it's amazing that you all come here and gather together and really rally around your family members and around yourselves for some of you, and it's just wonderful to be here and meet so many amazing people who are really just trying to do something that is very, very difficult. So I appreciate what Susan said about the fact that, especially in pediatrics, palliative care is just, it's a different perspective on a disease. And what my field is really trying to do is to meet you all very early, if possible, and really walk along a disease process with you. Try to help make sure that communication is as good as possible between whomever your specialists are, wherever they are, and your family, and vice versa. Help you advocate for the needs of your child. Help you feel confident in decisions that you may need to make. And obviously, everybody's a little bit different in how this affects them and when and what's going on in their life at the time. But sometimes even a decision like a G-tube is not easy. And sometimes families aren't sure what the best thing is for their child or themselves or whatever the situation is for their loved one. There's a lot of other things that come into living life when you have a chronic, complex medical illness. There is a lot of psychology in the way that you just deal with that every day in your life. There can be, so there goes into, there's emotional aspects. For a lot of people, there's a spiritual aspect of suffering and distress that can come when you're trying to just make sense of something that you probably were not expecting to have to ever encounter in life. And so what my palliative friends and I are trying to do is really take often a multidisciplinary approach. So often if you can access a pediatric palliative care team, you will find that there are not just physicians, but nurses, social workers, sometimes child life specialists, sometimes psychologists. Every team's a little bit different in its makeup, who really are going to try to come around you, learn about your family, where are the sticking points, where are the things that we can talk about and maybe make easier. What other resources can we put in place? And really just try to ease a burden, sometimes a little bit, so that you feel as confident as you can in whatever your medical environment is, that you are able to feel heard, feel supported, and just access the best medical care that you can for your child. That was a lot of words I just said.
Thank you.
Can I ask a question? How many of you have ever encountered a pediatric palliative care specialists at all? A few hands. Okay.
Yeah.
It does exist in other countries, definitely in Australia.
You know, I think even the pediatric provider community has a ways to go in our awareness of the appropriateness of connecting you early with families. So we have a team at OHSU now, and that was one of the most important things I learned from them. Yeah.
And I think that is, it is, we are all still learning together, and there is definitely sometimes just people don't even think about referring people or having those conversations. So it's certainly something that you can ask your specialists about, to try to access if you feel like that's something you at least maybe want to know more about, and find out what's in your community or whatever your closest children's hospital. That's where most pediatric palliative care teams live, but there are some that are spreading out into communities and exist to some level in more sort of regional or local hospitals. It's a growing field. So just like a lot of people don't know about PKAN, a lot of people don't know about pediatric palliative care.
I just want to add a comment from my own experiences with pediatric palliative care. They were able to help my family get some medical equipment that wasn't covered by insurance. Things like bath chairs, generators for your home if your power goes out. They have access to resources that have funds for the disabled community. So, and I think there's another family who connected with palliative care that was able to get some like acupressure or acupuncture to help just with the movements. Their palliative care is a phenomenal resource, and their focus is on really us as people, not as patients.
Thank you. That was very helpful. We have about 10 more minutes, and then unlike you all who probably don't have other specific sessions today, we did a BPAN session this morning. I told you we're doing this now, and we have a PLAN session yet to come. And so at the end here, I'm going to ask that you understand that the PLANers are coming in, and we're going to do another hour and a half with them. And so we are here, most of us are here for at least a few more days. Penny and I are here till the bitter end. We will have time to talk, but probably not right after this session. So we'll probably catch you sometime tomorrow. There's a training session, again, for the training physicians who are here. Some of you are already scheduled for that. Others may want to come in and meet with some of the docs then, but we'll have time to talk. So I just wanted to put that out. Are there other questions for now? Yeah.
I know that everybody's kind of centered on the CoAZ, but I believe when we met with you, you were talking about a parallel research effort for the gene therapy, and I know that you had mentioned L'Oreal, I believe.
Yeah.
And I believe that you've got a fund grant, something from the NBIA Association, but
Yeah.
how are we funding folks like L'Oreal? How do we
Yeah.
get her funding? Is she funded as part of Spoonbill?
Good question. Yeah, good question. Do you want to answer, L'Oreal, for where your funding is coming from?
So as a postdoctoral researcher, my main funding usually comes either from the person I'm working for, my mentor, a PI, or it'll come from an independent fellowship. So I'm on an independent fellowship. So my actual salary comes from a fellowship that funds just young academics, trying to make their way in the world. And so the project funding that I received that would then pay for what we do for the project. So that pays for all of the materials and the mice and all of the resources for that. And so those project funds that come in anywhere from a couple thousand to several tens of thousands really help to fund the actual specific projects. Does that answer your question?
Yeah. So there are two funds. One is the AISNAF, the Italian family group, collected funds from, I think, the three family groups, the German, the Italian, and the US family group. Somebody correct me if I'm wrong. Yeah, Veronica's shaking her head. Collected funds, and they sort of organized that one. including one for PKAN. L'Oreal competed for it and won. So those were your funds, your hard-raised funds.
Thank you. We're working on stuff
And,
now.
and the other was the American Society of Gene and Cell Therapy,
so the,
right?
another way we were able to get funding for PKAN gene therapy research is that my professional organization is the American Society for Gene and Cell Therapy, and they were funding projects in career development, and so that was gene therapy-based products for development of new treatments or new therapeutic platforms in gene therapy. So
Yeah.
there's a lot of different funding agencies out there that can help fund projects around, maybe they aren't specifically for PKAN, but for, there could be neurological disorder treatments or just for gene therapy platforms, and so we look to where who's funding and then apply for those funds.
Yeah. So it speaks to another point that I think is sometimes not so clearly understood, which is that the family organization provides project-based funding. Historically, it was around 25, then it went up to 35, 45. Now I think it's around 45 usually, 1,000. That's a lot of money. It isn't a lot of money in science because you have to not only buy the stuff to do the science, the supplies and the reagents and the mouse housing costs and the drugs and all the enzymes and all that sort of stuff, all of which is quite expensive. You have to buy the person. So in Europe, it's different. In the UK, it's different. The government does a lot of the salaries of the scientists. In the US, in the academic healthcare system, which is where OHSU is, where UNC is, Dr. Early is, we have to pay for everything. So one of the challenges that we face in our group and others face in their groups in the US is that $45,000 doesn't actually help. I can't do that unless I have somebody sitting on their hands waiting for a project to come by, and we never have that. We always have at least 50% more to do than we are really reasonably able to do. We're always pushing, always pushing. And so it's hard to think about, but you almost need like a bigger jump in order to get a person in, a new person who is dedicated to gene therapy, 100% dedicated to whatever it is, BPAN, gene therapy, whatever it is. You have to get a person who's dedicated to that, and then you need the supply money for that person to be able to do what they're doing. One of the challenges of these $45,000 jumps is that they don't get us the person. And so unless we have some other way to buy a new person, to add a person, we just can't keep piling projects on because we don't have the foundational support. Yeah.
I want to make one thing very clear. The Spoonbill funds have not been used for any salary of any person. So, that was a...
It's bought the compound, it's bought the development of the compound, it's bought the shelf life studies, it's bought the preparation of the formulation, it's bought everything that Penny and the others have been sweeping forward to get to this point, but it hasn't paid for people. And then the NBIA Disorders Association, from some fundraisers led by some of you in this room, put together a substantial donation that was dedicated to the database build. And so that bought the database build, not our people to do the database build, but to buy the database people to build it.
So our salaries, my salary, full disclosure, has never been paid by my university per se. So, it's a little known fact that in most academic institutions, we bring in our own salary. And so I can work on NBIA partly because I spend a fair amount of time working on Parkinson's disease. Don't tell the Michael J. Fox Foundation, but the only reason that I do as many biomarker studies for the Fox Foundation is so that I can, keep paying the bills that are not going to be paid for by Spoonbill funds, because we've been crystal clear that we're very much using that for very specific pieces of this project. And one question that I think sort of kind of came out of the last question is, so say we get to the end of this and, we have our big NIH grant and that helps and the Spoonbill funds help and we complete the COAZ trial and we still have money left over in Spoonbill, what's going to happen to that? I think we would come back to you, the community that raised those funds, and talk to you about exactly what you want to have happen to that. That's sort of a pie in the sky idea at this point, but I think it's a very real possibility that those funds would get used to pay for the next project, the gene therapy project, to support that. So
Yeah.
you would have an instrumental role in deciding what happened to any funds that were left over.
Or accelerating the trial in other countries, or maybe leveling the playing field in the US once a trial is done so that it's accessible regardless of your ability to pay, those kinds of things. There's lots of considerations. I think we have time for, yeah.
Yeah, I just want to say one thing. I'm Sue, and I'm a PhD, not an MD, as program just happened to be mistake me. Well, happened to be MD today. I just wanted to thank you all for sending all the samples. The blood requests, skin biopsy, whenever you come to us, we try to snap a tip of you. 2 years ago, we got boxes and boxes of samples, and they all come to me. And what we do is that when we see something promising or interesting, we don't just test on mouse. We test on human cell lines. We test on unprocessed fresh blood. So whenever a new idea comes up, we ask Allison and the clinical team, can I get some 10, 20 samples of blood? And she goes diligently working on emailing you guys, sending out the tubes. Could you please send the tube back? Stuff like that. But that way, we confirm things triple times, and that makes us give a very good confidence, an idea about data and how cells work. And that's only happening because of you guys and the patients and our clinical team. Yeah. And I just wanted to thank you. This is awesome community. I've ever seen it before.
Yeah.
Well, that's actually the perfect segue for me, because in room 5, our study coordinators and a very nice pediatric phlebotomist and another phlebotomist, our own Katrina, are all set up to collect samples, specifically from people with PKAN, because even though we couldn't start the clinical trial today as we had hoped, we can still be doing some of that legwork that we need to do. So if you haven't given us a blood sample in the last 3 months-ish, or if we haven't been sending you kits at home, if you haven't seen us in a while and you're willing to swing by room 5 when you leave here, or tomorrow afternoon, we're there, I think, from about 11 to 4. Not there, but in a different room next to the main conference room. We'd really appreciate sending a little bit of you home to our one lab tech, Dolly, who is still in Portland while the rest of us are here.
Thank you. Yeah. Did you have something, Marianne, or we're done? So I'm going to wrap it up. Again, we're here for the whole meeting. We will look forward to talking to you individually, probably starting tomorrow. But we do have, I think the PLAN session is right on the heels of this.
Yeah, it's literally
in this room. So thank you all. Thank you.